Hello, my name is Clifford Soloway, and I am a senior at Walter Panis High School. I would like to take this opportunity to essentially tell the tale of the research project that I recently submitted to the Davidson Fellowship Program. My research began when I was a freshman in my science research class. In this class, for the first 2 years, I was encouraged to choose my own topic to research further and read independently various scientific research articles related to that field. I knew from the very outset that I wanted to study something related to cellular metabolism. My interest in this field was derived from the class I took that same year, Living Environment, in which we learned the most basic structures of the process by which cells produce energy. Although my education as a ninth grader certainly did not go into the biochemical details of the process, it was simply the nature of metabolism that really sparked my natural curiosity. Simply the biochemical side that we did learn of converting glucose all the way into adenosine triphosphate via many metabolites along the way, and the protein side came together to produce something that I saw as almost magical. However, I did not want to study metabolism in a traditional sense. I did not want to solely contribute to the understanding of how it works in normal cells. I wanted to apply it to a new context. And it was in my school's science research program that I essentially stumbled upon an article describing the Warburg effect. And what I came to understand the Warburg effect was it was a phenotype observed in diverse cancer subtypes by which cancer cells rely heavily on glycolysis or anaerobic respiration in order to produce ATP. Now, this seemed counterintuitive to me because normal cells produce energy via oxidative phosphorylation, which produces around 34 ATP per glucose molecule, as opposed to the 2 ATP per glucose produced by glycolysis. And it seemed to me that cancer cells that are reproducing much more rapidly, would require that extra energy in order to sustain proliferation. However, I found through my initial study that there is a general hypothesis that glycolytic intermediaries form the basis for the necessity of the Warburg effect. And what I mean by that is glycolytic intermediaries, or the metabolites produced along the glycolytic chain, are able to be diverted into biosynthetic production pathways. And these pathways are able to produce amino acids and nucleotides that are essential to the formation of new cells or to reproduction, which is essentially the role of all cancer cells. However, the intermediaries of glucose oxidation do not serve a similar purpose. So there's a general hypothesis that the Warburg effect is necessary in cancer cells because it allows for the production of these various biosynthetic materials. So having established a central understanding of the Warburg effect, I sought to point my research in a new direction and give it some shape in order to lead into my own original research. And it was at this point that I reached out to Dr. Taro Hitasugi of the Mayo Clinic in Rochester, Minnesota, and by maintaining a dialogue with him, he began to shape the research process for me. He showed me that research is much more about the questions you ask than about the answers you find. And also in my dialogue with Dr. Hitasugi, my knowledge of the Warburg effect expanded, as well as my understanding of the field of post-translation modifications. Now, PTMs, or post-translation modifications, are a common regulatory mechanism seen in most eukaryotic cells. And it is a process by which the amino acid of a particular protein is altered via the addition of a chemical moiety. And changing the chemical structure of just one or more amino acids changes the way that amino acid correlates with other amino acids and how it interacts with the other amino acids on the same protein. And by changing this biochemical function, you change the conformation and the shape of the protein. So Dr. Hitasugi's lab was focused on studying post-translational modifications as related to various aspects of the Warburg effect. And by discussing his research with him and past research related with him, I decided to study more specifically the post-translational modification lysine succinylation. This is a relatively new post-translational modification. It was discovered only around 4 or 5 years ago, and it has not yet been studied in the cancerous setting. It has, however, been related to metabolism when it was shown that it affected the pyruvate dehydrogenase complex in addition to the succinate dehydrogenase complex. So my overlying question that I developed during this period was, how does the Warburg effect arise in tumor cells? There's already a general understanding of why it is necessary for cancerous development, but there is not a general decision as to whether it precedes tumorigenesis or if it is simply the byproduct of other processes occurring in the cell. So I sought to contribute to this overall understanding by studying whether or not lysine succinylation might be a contributor to the mechanisms of the Warburg effect in cancer. So from there, once I had established that underlying question and a more specific question, Dr. Hidetsugi and I designed a particular experiment by which I might elucidate that final answer, and I was able to go out and work in his lab for the summer before my senior year. I lived on my own in an apartment and essentially was able to experience the life of a real biochemist, a real researcher. I've spent every day in the lab doing my lab work, in developing and implementing the techniques that Dr. Hidetsugi was able to teach me, and it was from there that my research was developed and was summarized in the research report that I submitted. So the last aspect of my project that I would like to touch on is its significance. Now obviously I only studied one minor detail that was contributing to the Warburg effect, only one post-translation modification, lysine succinylation. However, this minor stepping stone indicates that the Warburg effect is a result of various processes. It is not simply a byproduct of other things occurring in the cell, but it is clearly the result of various changes that are occurring over the course of tumorigenesis. Furthermore, my analysis of both the post-translation modification and the metabolism of cells indicates the new wave of cancer research. In the past, most cancer researchers have concentrated on one of 8 cancer hallmarks of cancer. And the hallmarks of cancer pertain to everything from energy production to reproduction to protein regulation. And it has been through the understanding of each of these individual characteristics that set cancer cells apart from other cells that researchers have proceeded and have been able to develop treatment in the past. However, most of these hallmarks are relatively well understood now. There's a general consensus as to the reason for each of them and how they're coming about in cancer, or the beginning of an understanding of how they're arising in cancer. The next wave of cancer research indicates how they are related, how protein regulation affects metabolism, how metabolism affects reproduction. And my particular project showed an intertwining of protein regulation via post-translation modifications and cancer cell metabolism. So it is this mindset that we can only understand cancer if we understand how each of its characteristics are interrelated that adds significance to my work. And the final aspect of my research that is significant is simply the Warburg effect. The Warburg effect is a main difference between cancer cells and normal cells that represents a potential therapeutic target for the future and something that has already been used as a target for detection studies. The PET scan is probably the most common mechanism by which early stages of cancer are identified, and it functions by feeding the body with radioactive glucose, which will pop up as different from other glucose, so you can track which cells are taking in the most glucose. And cancer cells are taking in more glucose than normal cells because they're relying on glycolysis as per the Warburg effect. So the PET scan functions because of the Warburg effect, and that lends significance as far as detection to what has already been studied. However, as a therapeutic target, cells cannot survive without producing energy. So if you're able to cut off the energy production of cells, you will most likely induce apoptosis. As far as cancer cells go, they produce energy in a vastly unique mechanism, which represents a potential target for future drugs so that they would become more specific. Drugs that are able to target a particular focal point within the Warburg effect will only affect cancer cells and will have a vast specificity, allowing normal cells to thrive and to continue to survive, minimizing those side effects that are common to current cancer cell treatments. This has been a summary of my work. I would like to thank the Davidson Fellowship Committee for the opportunity to apply for your scholarship. I would also like to thank my mentor, Dr. Taro Hidetsugi, my teacher, Mr. Frank Tamweber, and my parents, and all those who have supported me along the way. Thank you very much.